Welcome back to IGCSE Science Academy, where science made simple and clear. Today we are going to solve the biology 0610 and 41 past paper. We are looking at the 061041 May-June paper. This is extended theory paper, which means it covers both the core and supplement material. We have 1 hour for 15 minutes to tackle a total of 80 marks. That's roughly 1 minute per mark, leaving us 5 minutes at the end of a final review. Before we dive into the biology, let's look at the logistic tools. Black or dark blue pen only. HB pencil is strictly for diagrams and graphs. Write your name, center number and candidate number in the boxes at the top of the page, as the boxes are mentioned over here. Write your answer to each question in the space provided. Do not use an erasable pen or correction fluid. Do not write on any barcodes. You may use a calculator. You should show all your working and use appropriate units. Here is the information. The total marks for this paper is 80. The number of marks for each question or part question is shown in the brackets. So these are the some instructions and information for the biology paper 4, which is theory for the extended students, and the code is 061041. Let's get started from question number 1. Now discuss each question along with their answers one by one. Here we have question number 1, part A. Figure 1.1 is a photograph of a fish. Figure 1.2 is a photograph of an amphibian, as you can see. The question for these two figures are: state two visible features that distinguish the fish in figure 1.1 from the amphibian in figure 1.2. This question carries 2 marks, so you only need to give 2 points. Here is the answer. To answer this, you need to look for physical structures present in the fish that are clearly absent or different in the amphibians. First is scales. The fish figure 1.1 has visible scales covering its body, whereas the amphibians figure 1.2 has smooth, moist skin. 2. Fins. The fish has prominent structure, fins, dorsal, pectoral, etc., while the amphibian has limb like feet or for movement. So these are the 2 points. Here is one more alternative point for you. You can also write this point instead of point 1 or 2. You could also mention the operculum gill cover visible on the fish, which is not present on the amphibian. So this is the answer for question number 1, part A. Part B, the statement is fish, amphibians and mammals are all vertebrate group. State the name of one other vertebrate group. So fish, amphibians and mammals are all vertebrate groups. State the name for one other vertebrate group. Is vertebrates are divided into 5 main classes. Since fish, amphibians and mammals are already mentioned, you can choose either of the following. One, reptile, for example snakes, lizards and turtles. Second one is bird, that is apes. You can use one of the from these two. Part C of question number 1, figure 1.3 shows the circulatory system of a fish. Figure 1.4 shows the circulatory system of an amphibian. Here are two figures, figure 1.3 and figure 1.4. Here is the question: describe the similarities and differences between the circulatory system of the fish and the amphibians in figure 1.3 and figure 1.4. So how you can write the answer for this, because this carries 4 marks, so you need to write 4 similarities and differences. Here, based on figure 1.3 and figure 1.4, here are the similarities and differences between the circulatory system of a fish and an amphibian. Similarities: both systems have a heart. As you can see, both have heart that acts as a pump to move blood through the body. Both have a closed circulatory system, meaning blood is contained within vessels and passes through capillaries at the body tissue. Third point, both systems include a ventricle and an atrium or arteria within the heart structure. Both have body capillaries where the exchange of gases and nutrients with the tissues occur. What are the differences? Number of circuits. The fish has a single circulatory system, one loop, whereas the amphibian has double circulatory system, two loops, as you can see. Heart chambers. The fish heart has two chambers, one atrium and one ventricle, while the amphibian heart has three chambers, two arteria and one ventricle. Third difference is gas exchange site. Blood in the fishes passes through gill capillaries, whereas blood in the amphibian passes through lung and skin capillaries. Fourth difference is blood flow path. In the fish, blood goes from the heart to the gills and then directly to the body. In the amphibians, blood returns to the heart after being oxygenated in the lung skins before being pumped to the rest of the body. Question D. Explain the advantages of the type of circulatory system in mammals compared with the type of circulatory system in fish. This question carried 3 marks and the answer is advantages of mammalian circulatory system as fish. Mammals possesses a complete double circulatory system with a 4-chambered heart which offers significant advantages. Higher blood pressure. In mammals, blood is pumped to the lungs and then returns to the heart to be repressurized before going to the rest of the body. This allows for faster blood flow to tissues compared to fish, where pressure drops significantly after passing through the gills. Number 2, separation of blood. Mammals have a complete spectrum that prevents oxygenated and deoxygenated blood from mixing. This ensures that tissues receive blood with the higher possible oxygen concentration. 3. Higher metabolic rate. The increased efficiency in oxygen delivery supports endothermy, maintaining a constant body temperature and a more active lifestyle. Part E. Explain how the structure of arteries and veins relates to the difference in the pressure of the blood transported by these vessels. This carry 4 marks and the answer is structure of arteries and veins related to blood pressure. The physical structure of these vessels is specifically adapted to the pressure of the blood they carry. Arteries high pressure, thick elastic walls. Arteries have thick layer of muscles and elastic fibers to withstand and recoil against the high pressure surges of blood leaving the heart. Narrow lumen. A smaller internal diameter helps maintain the high pressure needed to push blood to the extremities of the body. Veins low pressure. Thick walls since blood pressure in much lower by the time it reaches the vein. Thick muscular walls are not required. Wide lumen. A larger internal diameter reduces resistance to blood flow, helping blood return to the heart. Valves. Veins contain semilunar valves to prevent the backflow of blood due to the low pressure and the effect of gravity. Part F. Table 1.1 shows the names of some organs and the names of the main artery that brings the blood to the organ. Complete the table. Table 1.1 Name of the organ lungs, name of the artery that brings blood to the organ. You need to fill this. Second, you need to fill the name of the organ and the name of the artery that brings to the blood, that is renal artery, and here name of the organ. Third, liver, you need to name the artery that brings blood to the organ. Here is the complete table. Name of the organ lungs, name of the artery that brings blood to the lungs, pulmonary artery, Name of the organ kidney, name of the artery that brings blood to the organ, that is renal artery, name of the organ liver, and name of the artery that brings blood to the organ, that is hepatic artery. Here question number 2. This question is graphical question. Part A figure 2.1 shows the internal body temperature of a human and the external environmental temperature during 6 hours in one day. Here is the temperature and here is the timer. The straight line shows internal body temperature and these lines shows external environmental temperature. Question 1 of Part A. The internal body temperature range is from 36.4 degrees centigrade to 37.0 degrees centigrade. State the range of the external environment temperature shows in figure 2.1. So the answer from the temperature results figure 2.1 is state the stage of the external environmental temperature shown in figure 2.1. The range is from 28 degrees centigrade to 39 degrees centigrade. Calculation starting point at 0 hours is 28 degrees centigrade. The peak is 5 hours is 39 degrees centigrade. And this carry only 1 marks. Explain the result for the internal body temperature shown in figure 2.1. This carries 6 marks and the answer is explain the results for the internal body temperature shown in figure 2.1. First is homeostasis. The graph shows that while the external temperature fluctuates significantly by 11 degrees centigrade, the internal body temperature remains relatively constant between 36.4 degrees centigrade and 37.0 degrees centigrade. Negative feedback. This is achieved through negative feedback mechanism. Cooling mechanism. When the external temperature rises between R2 and 5, the body uses sweating, evaporate, cooling and vasodilation, blood flow to skin surface to lose heat. Then heating mechanism. When the external temperature is low, for a 0 hour, the body uses vasoconstriction or shivering to retain or generate heat. Figure 2.2 shows a cross-section through human skin. Here is the diagram. Figure 2.2. Here is the explanation of each part. Part A, that is temperature receptor, detects changes in skin temperature, sends nerve impulses to the brain for thermoregulation. B, sensory nerve ending. Here is the part B. Detects touch, pressure, transmits signals to the central nervous system. And here this the C, nerve fiber carries electrical impulses between receptors, effectors and the central nervous system. Part D, adipose tissues fat. Here are the part D. Subcutaneous fat layer insulates the body, stores energy and cushions or glands. Part E, erector pili muscles. Here is the part E. Contracts to raise hair, goosebump, trapping and insulating layer of air. And finally part F, which is epidermis. Outermost skin layer barriers against pathogens, ultraviolet light and water loss. Table 2.1 shows the names of some parts of the skin. This letter identifies the parts in figure 2.2 and its role in maintaining internal body temperature. Complete the table 2.1. Table 2.1 here name of the parts letter in figure 2.2 and role in maintaining internal body temperature. Table 2.1 human skin and temperature regulation name of the part. First is fatty tissues or adipose and the letter in figure 2.1 that is D. The role maintaining in temperature is in solution. The name of the part erector muscles and the letter in the figure is E. Contracts to make hair stand up to trap air. And name of the part temperature receptor and the letter in figure 2.2 A or B detect temperature changes. Question 3, Part A, Figure 3.1 shows a drawing of a root hair cell and Figure 3.2 shows a drawing of a palisade cell. First, let's talk about mitochondria. If you look at the Figure 3.1, you will notice the root hair cell is packed with them. Here is the question. Explain the reason for the differences in the numbers of mitochondria and chloroplast between the root hair cell and the palisade cell shown in Figure 3.1 and 3.2. So mitochondria and chloroplast, Figure 3.1 and 3.2. Mitochondria, and the answer is the root hair cell. Figure 3.1 has more mitochondria because it requires significant energy, ATP, for the active transport of mineral ions from the soil against a concentration gradient. The palisade cell has fewer because its primary role is not high energy active transport. Chloroplast. The palisade cell, Figure 3.2, has many chloroplasts to maximize light absorption for photosynthesis. The root hair cell has zero chloroplast because it's underground in the dark. Photosynthesis cannot occur, making chloroplast unnecessary. Part B, Figure 3.3 is a photomicrograph of a cross-section of part of a xerophyte leaf. Part A, Part B, Figure 3.3. Here is the question. Explain why the part labeled A in Figure 3.3 is a tissue. Here is the answer. Xerophyte leaf, Figure 3.3. Part 1, why A is a tissue. Part A, phloem is a tissue because it consists of a group of similar cells working together to perform a specific function, transporting sucrose and amino acids. Part Part 2 is described two ways the structure labeled B in Figure 3.3 is adopted for its function. Adaptation of structure B, that is xylem. First, lignified walls. The walls are thickened with lignine to provide structural support and prevent the vessels from collapsing under the tension of the transpiration stream. Two, hollow or dead cells. The cells lack cytoplasm and end walls, forming a continuous hollow tube for the efficient, unobstructed flow of water and mineral ions. Part 3, describe one way the leaves of xerophytes are adapted to their environment, and the answer is leaf adaptation. Second, stomata or thick waxy cuticles to reduce water loss by transpiration. Part 4, describe one way the roots of xerophytes are adapted to their environment. Root adaptation, long, deep roots to reach underground water sources. This question number 3, altogether carries total 11 marks. Question 4, Part A. A student investigated the effect of lactase on 3 different liquids: milk, lactose-free milk, sucrose solution. The student used an indicator to test for the presence of glucose. A sample of each liquid was tested before and after treatment with lactase. The indicators turn brown in the presence of glucose. The indicator remains blue in the absence of glucose. Table 4.1 shows the result of the test. Here is the table 4.1. Liquid milk, color before treatment with lactase blue and color after treatment with lactase brown. Lactose-free milk, color before treatment with lactase brown, color after treatment with lactase brown. Liquid sucrose solution, color before treatment with lactase blue, color after treatment with lactase blue. So question is, explain the result for the 3 liquids shown in table 4.1. So in enzyme investigation A, explain table 4.1 results. Milk turn brown after treatment because lactase break down the lactase in milk into glucose and galactose, and the indicator detects the glucose. 2, lactose-free milk already contains glucose, so it is brown before and after treatment. Sucrose solution remains blue because lactase is specific to lactose. It cannot break down, so no glucose is produced. Part 2. The student kept the solution at a temperature that was close to the optimum during the investigation. Using your knowledge of the effect of temperature on enzyme activity, explain why this was important. Part 2. Importance of optimum temperature. It ensures the maximum rate of reaction as kinetic energy is high. 2. Prevents denaturation if too hot, which could change the shape of the active site and stop the enzyme from working. Part B. As part of a balanced diet, some government recommends that children drink milk that has vitamin D added to it. First, suggest the dietary reasons for this advice. Vitamin D in milk, and the answer is it is needed for calcium absorption to ensure healthy bone and teeth development in children, preventing diseases like rickets. To describe what is meant by balanced diet. Balanced diet, a diet that produces all the necessary nutrients, carbohydrates, fats, proteins, vitamins, minerals, water, and fiber in the correct proportion for an individual's specific needs. This question number 4 altogether carry 11 marks. Question 5. Figure 5.1 is a graph showing the effect of temperature on the rate of transpiration from the upper and lower surface on a leaf that is provided with a constant supply of water. Here is the graph: rate of transpiration, temperature, upper surface and lower surface. Describe the results shown in figure 5.1. Answer to the question 5, part A. To describe the result shown in the graph, you should focus on the relationship between temperature and the rate of transpiration for both leaf surfaces. First, initial increase region X. As the temperature increases, the rate of transpiration increases for both the upper and lower surfaces of the leaf. This is because higher temperature provides more kinetic energy for water molecules to evaporate. In plateau region Y, at higher temperatures the rate of transpiration reaches a maximum and levels of plateaus for both surfaces. This indicates that temperature is no longer the limiting factor. Comparison of surface. The rate of transpiration is constantly higher from the lower surface than from the upper surface across all temperatures. This is typically due to higher density of stomata on the undesirable of the leaf. Here is the internal structure of the leaf. Upper epidermis, palisade mesophyll, spongy mesophyll, air spaces, lower epidermis, waxy cuticles, and guard cells. Question 6, Part A. Polio is a viral disease that can cause nerve damage in humans. In one area, polio vaccination began in 1957. Figure 6.1 shows the number of cases of polio in the area between 1950 and 1970. Number of cases of polio from 1950 to 97, and here the dark color shows the number of cases of polio in these years between 1950 and 1970. Part A. Calculate the percentage change in the number of cases of polio between 1950 and 1952 in figure 6.1. Give your answer to two significant figures space for working. So here, based on the graph provided in figure 6.1, here is the step-by-step calculation for question 6 part A. First, you need to identify the value from the graph. Number of cases in 1950, that is 220. Number of cases in 1952, that is 80. To calculate the change in the cases, subtract the original value from new value to find the difference. 80 minus 220 is equal to minus 140. Calculate the change in case, subtract the original value from the new value to find differences. Minus 140. Calculate the percentage change. Use the standard formula for percentage change. Percentage change is equal to change divided by original value multiplied by 100. Percentage change, you need to put the values minus 140, then 220 multiplied by 100, that is minus 63.3636%. Final answer is rounding to two significant figures are requested by the question -64% or 64% decreases. So the answer is 64% decreases and this question carries 3 marks. Then question 2, that is from question number 6 part B. Question 2 is explain how vaccination causes the result shown between 1958 and 1970 in figure 6.1. This question carries 5 marks and the answer is explain how vaccination causes the result shows between 1958 and 1970 in figure 6.1. The graph shows a dramatic decline in polio cases following the start of vaccination program in 1957. This occurs due to the following biological processes. Introduction of antigens. The vaccine contains a weakened or inactive form of the polio virus antigens. Immune response. These antigens stimulate the body's lymphocytes to produce specific antibodies. Memory cell. The body produces memory cells that remain in the blood for a long time. Rapid secondary responses. If the person is exposed to the actual polio virus later, these memory cells recognize the antigens and produce antibodies much more quickly and in larger quantities. Herd immunity. As more people become immune, the virus has fewer hosts to infect, drastically reducing the overall number of cases in the population as seen in the graph after 1958. Part 3, explain why the polio vaccine does not protect you from other diseases. This carry 2 marks and the answer is vaccines are highly specific because specific antigens. The polio vaccines contain antigens that are unique and specific only to the polio virus. Specific antibodies. These antigens stimulate the production of antibodies with a specific shape that only fits the polio virus. Complementary shapes. Other diseases have different antigens with different shapes. Therefore, the antibodies produced for polio will not be complementary to, will not fit the pathogens of other diseases. Part B, blood clotting helps to prevent some infections. Outline how a blood clot is formed and how it can prevent infections. This carry 3 marks. So how a blood clot forms? Answer is platelets. When a blood vessel is damaged, platelets release substances that trigger a chemical reaction. Fibrogens to fibrin. This reaction converts a soluble protein called fibrogen into insoluble thread of fibrin. Mesh formation. The fibrin threads form a mesh or net that trap red blood cells to form the clot. How does it work? Physical barrier. The hardened clot scab form a physical barrier that prevents pathogens like bacteria from entering the body through the wound. Prevent blood loss. It also prevent excessive blood loss, maintaining the integrity of the circulatory system. So this carry 3 marks. State the name of the component of blood responsible for transporting blood cells and the answer is plasma. This carry 1 mark. The question number 6 altogether carries 14 marks. Question 7, figure 7.1 is a flow chart showing the stage of eutrophication. Complete figure 7.1. Fertilizers enter a leg, then the construction of dish increases, then the growth of dish on the surface of the leg increases, decomposition then blocking dish from entering the water in the leg. Underwater producers die because they are unable to dish. Decomposition increases and bacterial decomposers use oxygen for dish, resulting in the death of organisms requiring dish oxygen. So here is the answer or the following the sequence of events when fertilizers enter a body of water. The first is the concentration of the fertilizers enter a lake. The concentration of nitrates or phosphate increases. The growth of algae on the surface of the lake increases. Number 3, blocking light or sunlight from entering the water in the lake. Then 4, underwater producers die because they are unable to photosynthesis. Decomposition increases. Bacterial decomposers use oxygen for aerobic respiration, then resulting in the death of organisms requiring dissolved oxygen. So this question carries 6 marks. Part B. A scientist obtained a sample of the bacterial decomposers and graved them in a flask. The resources available for the bacterial growth in the flask became limiting. The size of the bacterial population was estimated during the investigation, and these data were plotted on a graph. First, state the name of the expected shape of the population growth curve that would be drawn on the graph, and the answer is state the name of the expected shape of the population growth curve that would be drawn on the graph is sigmoid or S-shaped curve. Second is state the name of the initial phase of bacterial growth, that is lag phase. 3. State one factor other than a lack of resources that would cause bacteria to die during the death phase. That is accumulation of toxic waste products such as lactic acid or ethanol, change in pH levels to an unfavorable range. This question number 7 altogether carries 9 marks. That's all for today. In the next video, we will discuss another paper in detail. So stay tuned and keep preparing. Don't forget to subscribe.